We'll get going so that I don't have to cut you short at the end, if that's okay.
Sure.
Okay, good afternoon, everybody. Welcome back to the final session of the technology track over the last couple of days. I'm very pleased to introduce Gerardo Tauriello from Swiss Institute of Bioinformatics, who's going to be talking about Swiss model. And we were just saying Swiss model's got quite some history to it. So I don't know if you're going to say something about that, but Gerardo, over to you.
Thank you very much. Thanks to you for being here. Thanks for having me. So as you mentioned, I work for the Swiss Institute of Bioinformatics here in Basel in the group of Thorsten Schwede, heading the development team for Swiss model. And in this presentation, I'll show you how to do homology modeling of protein structures and complexes. So Swiss model, first and foremost, it's a web page. Here's the URL. This is how the front page looks like. As you mentioned, it has a long history. We celebrated 25 years last October in a little symposium where we also looked back at the history. You might have seen Thorsten's keynote on Monday in the 3D-SIG, where he showed a bit on where it came from. I'm not going to go into the historical part, so I'll just show you what Swiss model is today. And in terms of key features, first and foremost is a service to do homology modeling. So when you do homology modeling of a protein complex, what we mean by that is you have a couple of input sequences for a heteromer with two subunits. There will be two target sequences. It runs through a fully automated workflow with the possibility of user interventions, where it uses as a key input known structures of proteins that are homologous to your target sequences, which we call the templates, all right? And at the end of the day, you get out the predicted 3D structure, which is what we would call the model. So just to get the terminology cleared. So apart from being a homology modeling service, the second key feature that we write on our front page of the web page is that the purpose of the server is to make protein modeling accessible to all life science researchers worldwide. Sounds great. What we mean by that, with accessibility, meaning it should be free to use. There shouldn't be any licensing restrictions on the models that are produced, so you can do whatever you want with them. And we want it to be fast, so a key property of our system is that it produces results in a couple of minutes, while still trying to be as accurate as we can possibly be in that amount of time. We want it to be responsive, reliable, easy to use, or in short, it should just work, all right? And at the end of the day, we focus on having biologically relevant models, meaning we want the complexes in their correct quaternary structure, and we want to have accurate quality estimates on the models so that you know what you could possibly do with the structure that you get out of it. It's also heavily used. So to give some numbers, over the past 12 months, we processed roughly 3.2 models every minute. And over the past 4 years, we had over 2,000 citations every single year. Now, I'll walk you through the demo-like how it would look like to model a protein complex in Swiss model. So once you start a modeling project, what you see is mainly a big input field where you can enter the protein sequence. Or if you don't want to enter the full sequence, you can just enter the UniProt the UniProt code. So in this case here, this is the code for the alpha subunit of hemoglobin of the sperm whale. It's a hemoglobin which doesn't have a known structure yet. And as soon as you validate this, it gets translated to the full amino acid sequence, and you get a button which allows you to enter an additional sequence to say, hey, this is a heteromeric protein. It has multiple subunits. So I can click on that, enter the beta subunit of hemoglobin of the same organism. I get two sequences that I can use to start my modeling process. I have two options. I can search for templates and choose the templates myself that I want to use for modeling. Or as I will do for this demo, I can just say build model, all fully automated, basically the worry-free option. So once I start the modeling process, it will tell me about the progress, what's going on. Here it's going through the template search part where we identify what structures we could use for modeling. And also you get a stable URL right from the start, which you can bookmark and send to your colleagues that will work once the modeling is finished. And Once it's done, for this specific example, when I tested it last week, it took roughly 3 minutes, just to have an idea. What you're presented with at the end is the model results. You get a model structure in the correct oligomeric state in this case. It's a dimer of dimers, 4 chains, 2 subunits, just as you would expect from hemoglobin. And it discovered that fully automatically. We have a big focus on model quality estimate, so the whole middle part is for that. We have a 3D viewer, so it's a modern web page. It has interactive features. You can rotate it, of course. You can hover over the residues. It will highlight where you are in the sequence view, and vice versa. So it makes it very easy to navigate through the structure of the model. Now, for the remainder of the talk, I'd like to focus on 4 elements of Swiss model and its surroundings. One being the template selection feature. It's a reasonably recent addition that we did to automatically select the best quaternary structures, and tell you a bit about that. Our modeling engine, which once you selected a template, generates the final model. We open sourced that last year, and I'm going to show you that too. And our model quality estimation tool, we've been continuously improving that, and we believe that by now it's really a best-in-class error estimator. And I'll show you that. And finally, once you have a useful model for research, I'll show you what you can do with it, how you can archive it for further use. Now, for the template selection, first of all, let me show you how it looks like, how the template list looks like. So the result of the template search is essentially a list of possible structures you could use for modeling. So each of those entries has a PDB ID, so it has a known structure. It is homologous to the target sequences that the user asked for, and we were able to align it to it. So it comes with an alignment ready for you. You can select multiple ones of the templates if you want to compare them. It has a 3D viewer, et cetera, et cetera. In the table, you get 2 main numbers, main estimators that should guide the user in terms of what's the expected quality if I was to use this template for modeling. And we split it up in 2 separate parts. One is the GMQE, which tells me something about the estimated quality of the tertiary structure, so how accurately I can do each single chain. And the other one, which sometimes can be actually surprisingly complementary to it, is the QSQE, which tells me something about the expected quality of the quaternary structure. I'm going to tell you something about the QSQE on the next slide. So what we did there is a machine learning approach. We trained an SVM with various features that we can extract from the templates that we find in a template search. We have features related to sequence properties, to conservation, to amino acid composition, or simply what type of quaternary structures we see in the list of templates. And all of those get fed into the predictor and result into this QSQE, which was trained to be as good as possible in ranking templates in terms of choosing the best quaternary structure. 2 of the intermediate results of the features we also display to the user. That's on one hand a PPI fingerprint that tells me something about the conservation. It's based on the observation that interface residues are more conserved than surface residues. And based on that, we generated a score which results in the plot and also results in a couple of features that are fed into the SVM. Plus, in terms of the consensus of the quaternary structures, we display to the user in a tree view what kind of quaternary structures we found. In this specific case, it's for a single target for a single target sequence, which appears in the PDB in homologs that have as a monomer, as a trimer, as a hexamer, as a dodecamer, and it's not immediately obvious which one to choose. Now, once you selected the template, it gets fed into our modeling engine. It's called Promo3. It's an open source library. It has a C++ core, so it should be fairly fast. It has a Python interface around it, so it should be fairly easy to use. And you can get it at the URL written there. It supports Docker and Singularity, so you can install it fairly easily. It can do all the steps from basically you start with an alignment and with a template structure, and it can do all the steps needed to get to a final full atomistic model of the target protein. It has a very simple command line interface. So basically, this is all, if you have a FASTA alignment and the PDB file for a template, this is all you need to generate your model. But if you want to customize it and go deeper into it, you can also use the powerful Python interface that comes with it. This is essentially the exact same thing as the command line interface, the code snippet shown here. Just I expanded the functions a bit so that you get a couple of functions where you could add additional parameters to modify the way the various steps are being performed. Now, once we have a final model, we want to add model quality estimates on top of it. The idea there is like a thing that Thorsten keeps on saying in all his presentations, and it's very true, all models are wrong, but some are useful. So we want to basically help the user make this decision, put some number on the potential usefulness of the model. And we believe that a key to do that is to provide the user with accurate local quality estimates. So what you see there in colors and also in the plots is how accurate we believe that at each residue, how well we capture the neighborhood of this residue. So it's basically trained if I knew the correct solution of this protein, and I was looking at the residue and all its neighboring and all its neighborhood, how many of the interatomic distances do I capture correctly? And if we think that I capture all of them correctly, it will be blue. If I think that we don't capture them very well, you'll get orange and the lower value. Also, this has been open sourced sometime this year. It's also available as a standalone web service, so you can just upload your own model structures and see what comes out of it. Now, I'm saying that this is supposed to kind of predict what it would be if I knew the correct structure. Of course, normally I don't know the correct structures, with the exception of benchmarking efforts. So we're heavily involved also in benchmarking efforts, and the Cameo benchmarking efforts tries to measure exactly what we try to do here, in a sense, how well can we distinguish good residues from bad residues. And it's measured with ROC AUC and the precision recall AUC. And I show here the results of QMINDisco compared to a couple of other services that are around. This is publicly available data. You can go on the Cameo webpage. You will get the exact same plot. This is over a time range of 3 months, which 3 months meaning that it has scored almost 3,000 models in this time range. It also participated in the last round of CASP, where you have a kind of a similar problem set up on the global scale. So basically, how well can we classify good models from bad models? That's what you see in the table there. And you can have different metrics that tell you what a good model is and what a bad model is. So depending on which metric you use, you measure different things. The LDDT and CAT measures are the ones that for us are more interesting and also for our users. So we optimized for that. So hence, we were fairly good at that and not so good at other metrics. An important feature of it is also, since we aim to provide results in minutes, QMINDisco needs to be very fast. So it's also one of the fastest model quality estimator out there. The models, I've shown you how to run through the whole interactive pipeline. Since it's a fairly fast system, etc., we also have a repository where we keep proteomes of selected organisms updated weekly. The result of that is here I show one entry. So each UniProt ID has an entry in our repository. If we have a model, we display it. We also display annotations taken from UniProt, and you can map them on the three-dimensional space thanks to that. It's also, if you go to the UniProt webpage and we have a model on the repository, you'll find a link on the UniProt webpage. We're also linked from the RCSP webpage. So you can get to our models fairly easily from many different places. Now, a second problem is once you have a useful model and you use it in your research, what do you want to do with it? Like you wrote a manuscript, you refer to the model that you have, you will describe kind of what you did to generate it, but you need to store it somewhere. With experimental structures, you will put it in the PDB. With hybrid structures in PDB-Def, and if you have a theoretical computational model like homology modeling or de novo modeling, the solution is called Model Archive. That's also run by us. where you can deposit it. You get a stable DOI as a URL to go to your model. You can add any meta information that you want on how exactly the model was performed. We usually ask also the depositors to add some quality estimates because we think that's important. And the whole web page is searchable, findable, and all the things that you expect from following fair data principles. Now, before I conclude, I'd like to acknowledge the people that make all of that possible. You see on the right-hand side the current team members. Most of them are also in the conference, so you can grab them and ask them questions of anything you would like. Plenty of former team members that have contributed various parts of the softwares, and all the funding agencies that helped make all of this possible. Now, to conclude, what I've shown you is homology modeling of protein structures and complexes, how this is done in Swiss model, the Promote 3 toolbox that you can all download and play with, Cumin Disco that you can all download and play with also, or use as a web service. And I hope I convinced you to deposit your models in Model Archive if you want to have reproducible science. Now, in terms of next steps, there's two parts that I want to kind of throw into the audience to see maybe also what you guys think about it. Is in terms of extending the scope of Swiss models. So two things we will kind of be interested in doing in the future is, on one hand, we see that for many people the model structure is a first step for further analysis. And we'd like to see what are the very common further analysis that people do, and whether we can embed it into our web service directly so that people don't even need to download the model and use another tool, but they can just do it comfortably in the browser. The other part is that we've seen an increase of usage of our service via scripting. So some people have gone through the trouble of scripting access to our web page, which sometimes we need to restrict because it's kind of it causes problems to the web service when people do that. So if you want to do that, if anyone in the audience wants to do that, please write us an email instead of just bombarding us with requests. But to make this also more workable, we want to extend access to our web services with APIs. And one of the prototypes we already have is a collaboration with the CAF service that Ian is going to tell you more about the CAF service later, actually. And we want to extend on that, having more possibilities to access. So anyways, yes, we're very happy to hear you, any ideas that you have. I'm very happy to hear any questions that you have. And thank you very much for your attention.
Protein sequence which has template for, like, I have a protein sequence for 500 amino acid long and I get a template for 200. So how Swiss model deals with the rest?
All right. Okay. So at the moment we don't try to use information from multiple templates to extend coverage. So if there is template information covering several parts of your long protein, we produce multiple models.
Yeah.
So we basically just say we don't have any reliable way to figure out how the domain orientation is, so we're not even trying. And essentially it depends on how the domains interact with each other.
Yes.
For many purposes, we observed that looking at the single domains is enough. There are some rare cases where some template results get hidden somewhere down the list and you don't see them for some parts of the proteins. So for very long proteins, we usually propose that people can submit them separately for separate regions of interest. Sometimes that can give better results. Most of the times it works fine with the full sequence, but in case that doesn't work, splitting it up manually can help.
Yes, that can be done when you have multiple template. But if you have no template for like 200, so does it go for fold prediction?
So we don't try at the moment to do de novo predictions of parts where we have no template information at all. simply because so far the quality of the models that came out was, I mean, okay to capture the correct fold, but not good enough for an all atoms structure for variant interpretations or this kind of use cases. This seems to be changing also, so this might come back in the future. But at the moment, for cases where there's no template information, we usually tell people to use Robetta, ITES, or
sure.
any of the usual candidates.
Yeah, okay, thank you.
Thank you very much. So I'm trying to keep exactly on time so that if people want to switch between sessions, they could do so. So, Eric, is Eric here? Yo, okay, because we've got his slides. We haven't got Eric. Okay, sorry. So you're Timothy. So this is Timothy Reynolds from Baylor University. So Timothy, welcome. And just while you're going up on stage, the title